All right, 8th grade. We are going to talk now about how to analyze data, particularly with respect to using a regression analysis. So there's some key questions to ask whenever you're performing it. The first off is, what kind of variables are we dealing with? Are they categorical or are they continuous? Remember, if you're dealing with an IV, which is categorical, and a DV, which is continuous, then you're going to want to use a bar graph. And then you're going to do an error bar analysis, where you put in a standard deviation above and below the mean. However, if you've got two continuous variables, in other words, your independent variable is continuous and your dependent variable is continuous, then what are you going to draw? A scatter plot, okay? XY scatter plot. That is the graph that you're going to use if you're dealing with an independent variable that's continuous and a dependent variable that's continuous. And that is exactly what we're dealing with respect to canopy coverage and soil temperature. Canopy coverage is continuous, as is soil temperature. So we are going to be making an XY scatter plot, which means we can't use error bars. We're actually going to be plotting points on the graph so that then we can evaluate whether or not there is any kind of trend in that data using an XY scatter plot. So another question to ask is there any relationship between our variables? Okay, that's the number one question that you want to start looking at. So let's say we have some data and we've plotted that data and we see that suddenly we have this trend that seems to be heading upwards. Right there and then, hopefully you can all see that there seems to be some kind of trend in the data. However, you might have another data set whereby the data is spread out so randomly that you can't really tell whether there really is any significant trend whatsoever. Now, from a qualitative standpoint, you can kind of use words to describe that, but what we want to do is we want to try and quantify that. So that leads us to the next question, which is how strong is that relationship? And this is where we're going to perform a linear, a sort of regression analysis using a regression line. Now, more often than not, we are going to probably use a linear regression. For more advanced students, you might want to look into mathematical concepts that might be at the heart of the problem, and as a result then choose maybe an exponential analysis or a logarithmic or whatever it is. But what we're going to do is we're actually going to use a linear regression line to, that's linear regression line, to essentially quantify how strong the relationship is within the data set. So what that calculation does, and Excel is going to do that for us, what it does is it looks at all of the data and essentially it tries to fit a line where the distance between all of the points, that distance, 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 where the distances, and let's say each of those distances is written as d, where the sum of all those distances is small. By the way, this thing right here means sum of. So the sum of all the distances is small. And there are calculations that we could do as human beings, and when you get to college you might learn those, but for now we're just going to use Excel, and it's going to do it for us. And so then we're going to have a line that's put in there. The important thing to do when you've actually done that is to ask it to show what's called the R-squared value. Now the R-squared value is essentially our statistical number, probability, that any future point, so I take new data, and let's say new data is the brown, and the new data, it's the statistical likelihood that that new data is going to fall on that line, okay? So in other words, if we have an R-squared value, which is really high, or the highest it could be is 1, because that means there's a 100% chance, but if the R-squared value is close to 1, then it's really strong relationship, very strong. That's not going to fit, is it? Oh, you get the idea, very strong. If it's very small, in other words 0, then it means that there is no relationship between the two variables. Now, at the same time, you need to consider essentially how complicated the system is. with physics in a controlled experiment, similar to what we were doing last year in 7th grade, there aren't many variables that are going to affect the experiment because as a scientist you're able to remove those variables. Okay, you have lots of control variables. Controlled. And so the system is not too complex. If we're dropping a ball of different diameters and all of the balls have similar materials or the same materials, then we're removing variables from that and we're essentially making the system very, very simple. And so that means that in order to really state that there's a strong correlation between the two variables, you need a pretty high R-squared value. But as we head down here, you will see the complexity of the system increases. Consider biology, all of those genetic mutations and variations that affect living things. If we consider social sciences, all of the factors that go into that. And when we think of social sciences, we're thinking about economic theories, okay? So when we think about the stock market, consider all the variables that affect the stock market, from bioconfidence to politics, global trade, individual company issues. All of those factors are going to affect that, and therefore, looking for any meaningful relationship between variables means you actually need a smaller R-squared value. Because the system is so complex, you don't quite need the same R-squared value. For us, we are generally going to be dealing in this range here, chemistry and biology, because we're doing chemistry in terms of temperature. But you might also say that's physics, actually. But we're also then dealing with biology in terms of canopy coverage. And so actually heat conduction is probably physics as well. So how the sunlight travels down towards the soil, that's heat conduction within the soil, radiation between the photons getting down and hitting the soil, so that's physics. So we're looking for a sort of R-squared value in and around this range. But remember, consider the complexity of the system. It's not a hard and fast benchmark. When we're looking at the system that we're dealing with, we're talking about many, many environmental factors that are going to affect our data at Drumlin Farm. And it will be up to you to explore all of those different factors. So what other factors might be influencing the data? Well, it could be the various different variables that were not controlled. It could be procedural. In terms of the equipment we were using, or the different students that were performing the experiment, and how precisely each student was performing that experiment relative to other students. But it then also could be errors. Wow, I can't spell. It could also be errors in the experiment that might affect the data. And that's what we're going to explore next. So, as we look at this particular XY scatter plot here, we see that on the one axis here we have hours of sleep. That's our independent variable, and the dependent variable here is the percentage of correct answers on a test. And what we see is we have a linear regression line that's being put in here by the computer or the scientist, and they have posted the R-squared value. Remember, the R-squared value is the probability on a scale of 1 to 0, the probability that any future point will fall on this line. And as you can see, the probability here is very, very low. Remember, when we're changing this into a percentage probability, we need to shift that decimal point 2 times. And so we're dealing with a 0.018% probability that any future point will fall on this line. That's pretty small no matter what branch of science you're dealing with, okay? But look at this point over here. That point over there seems very skewed from the rest of the data. And consider the amount of sleep this person had. This person had 1 hour of sleep, which is very, very different from the rest of the group here. So let us say that this could be called an outlier. Outlier. Now, some of you will have heard that term before. Essentially, it's a data point that seems really off from the rest of them. And maybe something weird was going on there. So let's have a look what happens if we remove that outlier. What we find is if we remove that outlier from the data, notice that it's actually gone. We actually had to remove it from the data set. Otherwise, Excel is going to continue putting it in there. What we see now is we actually have increased our R-squared value here. Now, we have to be very careful when we do this as scientists, because we don't just want to start removing points so that in order to increase the R-squared value. That is seriously manipulating the data to get the results that you want. But what you do need to consider is all of the variables that might have affected the situation. For that particular student, there might have been just a fluke on the exam, or they were very well prepared and so they didn't need the sleep. Consider the data set size. You know, we were only dealing with, by the looks of it, about 1, 2, 3, 4, 5, and 6, 7, 8, 9, 10, 11, 12 data points there. And so, you know, removing one outlier doesn't seem all that unreasonable from this data set. But as we see, we've increased the R-squared value. In other words, we've increased the probability that future data points are going to fall on this line, as, again, excluding the outliers. So are there other ways to analyze the data or graph the data? That's a key question here. And it's something that you're going to have to consider when you do your final project at Drumlin Farm. In this particular experiment with the soil and canopy coverage, there are ways that we could change the data sets, and I think if you probably talk with your teacher, they could probably help you out with that. But it's always important to consider all of the data that you have. I have already had a couple of conversations with people who ask whether or not we're going to have multiple independent variables. And the question that I pose to them is, well, it depends. We're going to try and collect as much data as we can out there in this field study. And that's the difference, again, between a field study and a controlled experiment. Remember, in this field study, you need to collect as much data as you can because you never know what's going to impact the results. Let's take an example here. Let's say that we're looking at tree distance here, and we have a transect set up, and we do soil samples, or even we do temperature readings at linear distances away from this tree. And as you do that temperature analysis, you might take out other data from each of those points. You might look at the soil color. You might look at the soil texture. You might be in different habitats. You might be in different forests. You might have different species of trees that you're going around. And again, a lot of this is going to come down to the kind of experimental design that you've set up. And still, the idea of limiting your variables is very important, but you know, there's no way to control everything in a field study. And so being able to capture all of that data as you go around is going to be really, really important, because the more data you have from the field, the more ways you can try and interpret that data and try and find meaning behind your results. Let's consider these three sets of data, all from the same experiment. You can see that each of these experiments is investigating the water pH against the nitrate levels in the water. ppm means parts per million. And you can see that we've gone and sampled from 3 different ponds, which is reasonable because we want to not just focus in on one pond because there might be an issue with that pond. So we want to go and look around at all sorts of different ponds. As we graph the data from each of these different ponds, we can see very little correlation between Pond A and Pond B data, but Pond C data seems to have a pretty decent sized R-squared value. That's a 25% probability. Now, is it a good R-squared value? No, even social sciences shoots for more than 35%. But it's definitely a stronger correlation than the other two, which had a meager 1.5% probability of falling on the line. But why separate the ponds out? Well, obviously each pond is very different, and so the variables affecting each pond are going to be different, and so it does make a lot of sense to look at each pond individually. But what happens if we were to actually combine the data into one graph? Now what we find is we see a pretty clear trend that even without the regression line we can actually start to identify. We see that positive trend upwards, and I can already identify an outlier here, which if we remove that one outlier from the group, we might actually increase our R-squared value even further. But again, I wouldn't just remove it for the sake of removing it. I would remove it after considering all of the other issues with that data point. Were there any errors with respect to this data point? Is there any other qualitative data that I can use to help justify removing that point from the data set. Because if there is, then I could potentially remove this data point, again run the regression analysis, and this, for all we know, could be up as high as 0.6. And that is a pretty significant, meaningful relationship between the two variables in an environmental science field study. Again, what this R² value is doing, it's essentially quantifying the strength of relationship between our two variables, water pH and nitrate pH. And so in an analysis of this data, the scientist could quite confidently say that there is a fairly strong correlation between water pH and nitrate levels within ponds. And that R² value then is at about 0.55 or 0.6%, again, depending on what we do with that particular outlier. There are other graphs that we could draw as well. I mean, you saw that we had another independent variable, which was the different pond. And notice that that independent variable was categorical, right? The different ponds are categorical variables. And so what if we graphed the data of average pH and average nitrate levels? Notice that, of course, we're doing the averages here in our bar graphs, whereas we didn't do the averages in the XY scatter plots. We wanted all of the data points there in those XY scatter plots because there were no categories to average in the XY scatter plot. But here we have a category that we can average, and that's the pH and the nitrate levels of each pond. And of course, with the average, we put the error bars in there, and we can run an analysis of this, where we can say, look, these error bars here, they don't overlap, and so pond B has a significantly higher, what is it? It looks like the nitrate levels. No, that can't be it. That's going to be the pH. So the oh yeah, I can see the little boxes there. So the average pH is significantly higher in pond B than in pond C and in pond A, and the average pH level is significantly different. I'm sorry for my wavy line, but I think it's still significantly different from pond C and pond A. And so all of the ponds had very different pH levels. The nitrate levels, well, C and A, they're sort of starting to overlap a little bit. And I might want to zoom in on here, but B and A definitely look different. And A and C still look pretty different. And so again, we can compare these where we see that the highest pH level here is showing the highest nitrate levels. And in that I mean pond B. The highest pH levels are showing the highest nitrate levels. And in pond A, the second highest pH levels are showing the second highest nitrate levels. And moving on to pond C, the lowest pH levels are showing the lowest nitrate levels. So that's a whirlwind tour of analyzing data, and analyzing data using a regression line in an XY scatter plot using continuous variables. It's also a whirlwind tour of using your logical mind as a scientist and trying to look at the data in lots of different ways. And in this particular lab with the soil conductivity, with the soil temperature, we're giving you the data. We're giving you the graph with a regression line already on it. And so you can hopefully look at that regression line and now understand a little bit more about its significance and apply it to your lab report.